Good afternoon, ladies and gentlemen. Welcome to today's biology practical session. We will examine the microscopic architecture of plants and animal cells, investigate the structure of a bony fish gill, and perform essential food tests to identify key nutrients. By the end, you will clearly understand how these cell structures differ and how basic food tests reveal the nutrients present in our daily diet. Each specimen and solution has been carefully selected to deepen your understanding of cell biology, respiration, and nutrition, core topics in biology practical across all examination boards in the world. As we normally do in my biology practicals, let me give you the confidential or the requirements for the biology practical paper of 1984 EACE, meaning this examination was done by Kenya, Uganda, and Tanzania. So it was a tri-national examination. So in question 1, we require a permanent slide of a plant cell labeled specimen K1, permanent slide of an animal cell labeled specimen K2. In question 2, you are required to have a gill from a bony fish labeled specimen K3. And lastly, for question 3, you're required to have the following: glucose solution labeled solution X, starch solution labeled solution Y, Benedict solution, dilute hydrochloric acid, sodium bicarbonate powder, and means of heating. So let's come to the questions you are supposed to answer. In question 1, part A, you are supposed to examine specimen K1 under the microscope, and you draw 2 cells. Label all the parts and you state the magnification. Part B, you examine specimen K2 under the microscope and draw 2 cells. Label all the parts and state the magnification. Part C, from the observations you have made under the microscopes, from the drawings, list 3 similarities and 3 differences between specimen K1 and K2. The best thing is to make a table of comparison where you have specimen K1, specimen K2, and then you have similarities, and then you have differences. Part D, giving a reason, identify specimen K1 and K2. So, specimen K1, give a reason, and specimen K2, you give a reason after identifying. We now come to question 2. Specimen K3 is an organ from a bony fish. Part A, using a hand lens, study the specimen. Draw and label all the structures of the specimen. Part B. State the function of each of the structures labeled in 2A above. Use the table below. The table has structure and function. And finally, in part C, describe the main processes in the bony fish which involves specimen K3. And finally, let's come to the questions of question 3. Identify solution X and Y by carrying out the food tests as indicated in the table below. So you have the table, the test for reducing sugar and the test for non-reducing sugar. We have solution X, Y, and then solution X, Y for non-reducing sugar. Treatment, observation for X and for Y as far as reducing sugar are concerned, and then observations for X and Y as far as non-reducing sugars are concerned. And finally, conclusions for solution X, solution Y for reducing sugars, and finally, conclusions for X and Y for non-reducing sugars. Roman 2. Which of the two solutions, X and Y, would you recommend for a person who needs an immediate source of energy? Give a reason for the answer. So we have the solution to be recommended, and then the reason why you recommended that solution. So we are now looking at the answers and the way it was marked. And we start with equation A, drawing two cells of K1. You can see the cells there, where we have labeled in one of them cell wall, cell membrane, cytoplasm, and the nucleus. You are supposed to get 5 marks, so there are 4 marks for labels and an accuracy mark. And the accuracy mark is awarded because of the following reasons. We have outline is continuous, nucleus is placed at side north center, no shading except nucleus only, cells must appear elongated, then double line, more than one cell, and there is no use of ruler when drawing. The magnifications range from 20 to 225. Instead of putting the X in front of the number, you can just say, if I take, for example, X50, you just say 50 times. You get the correct mark. In 1B, the drawing of specimen K1 has been done, and what I'm showing here is a diagram of an animal cell, so that, and because we know K1 is an animal cell, and therefore The rebels are small temporary vacuole, cell membrane, cytoplasm, and the nucleus. The three of them form what we call protoplasm. Then we have glycogen granules, and then we have oil droplets. The magnification still ranges from what and given. So we have just drawn two cells, but still on this slide you can see a slide of a number of cells, but we have picked one for this question 1b. Now let's look at part C after the drawings. 3 similarities and 3 differences between specimen K1 and K2. For similarities, they are eukaryotic, they have cell membrane present, they have cytoplasm present, they also have inclusions or non-living objects, and finally both of them have organelles present, which are the living objects. Differences. Specimen K1 has large permanent central vacuole, while specimen K2 has temporary small vacuoles within the cytoplasm. For specimen K1, we have chloroplast, but for specimen K2, we have no chloroplast. There is cellulose cell wall in K1, but no cell wall in K2. And finally, specimen K1 has a regular shape, while specimen K2 has irregular shape. Part B of question 1, giving a reason, you identify specimen K1 and K2. Specimen K1 is a plant cell, and the reason is it has cell wall enclosing a cell membrane. It is regular in shape. Specimen K2 is an animal cell because it has only cell membrane and it is irregular in shape. Now let's dive into question number 2. Drawing and labeling all the structures of the specimen. And the drawing is there. And we have even shown where you put the marks for accuracy. All right. Then you have the rebels from the top. You have gill filament, gill bar, or what you can call it, gill arch, and gill raker. For rebels, you have 3 marks, and for accuracy, you give 2 marks to give a total of 5 marks. So let's come to the way you are supposed to have marked this. Note, mark accuracy 1 and accuracy 2 independently. labels should be given correct names or should be correctly spelled. So for accuracy, the first mark, you should have continuous outline, all parts of gill drawn properly, and no shading. And for you to get the second accuracy marks, because we have seen they are independent, all the three parts drawn proportionally in correct sizes in relation to one another. The curvature of gill bar must be shown. Gill rakers should not be close to each other. Question 2b. Stating the function of each of the structures labeled in 2a above. We have a column of structure and a column for function. So for the structure gill filament, the function is to increase surface area for gaseous exchange, and another function is to reduce the distance through which gases diffuse because it is thin. For the gill bar, or what you call the arch, the function is to provide firm attachment for gill filament, or you can say provide firm attachment for gill rakers. And finally, the structure gill raker is to prevent solid particles from reaching gill filaments. Describing the main process in the bony fish, which involves specimen K3. The first point is water intake and flow. The fish opens its mouth to allow water to enter the buccal cavity. It then closes the mouth and raises the floor of the buccal cavity, increasing pressure and forcing water over the gills into the opercular cavity. 2. Second process is the gill structure and function. Each gill consists of a gill arc with gill filaments and lamellae. The lamellae are thin and richly supplied with capillaries, providing a large surface area for gas exchange. Another process is diffusion of gases. Oxygen dissolved in water diffuses across the thin epithelium of the lamellae into the gland in the capillaries. Carbon dioxide or carbon 4 oxide from the gland diffuses out into the water and is expelled through the operculum. The other important process is the countercurrent flow mechanism. In this mechanism, water flows over the gills in the opposite direction to the blood flow in the capillaries. This maintains a concentration gradient, maximizing oxygen uptake and carbon dioxide or carbon 4 oxide removal. The next process is efficiency adaptations. The gills have numerous lamellae for increased surface area. Thin membranes and rich blood supply enhance diffusion efficiency. And finally, final expulsion. Deoxygenated water exits through the operculum, completing the ventilation cycle. Question 3. Identifying solutions X and Y by carrying out the food test as indicated in the table. So test for reducing sugar. For solution X, you add the Benedict solution whole, followed by you heat or you warm to boil. And boiling is not a must. You can just warm. For X, color changes from blue to green to yellow to orange, reducing sugar present. For Y, no color change, reducing sugar absent. For non-reducing sugar test, for X and Y, you are adding each and dilute hydrochloric acid, heat to boil, cool under cold water, and sodium hydrogen carbonate, and then add the Benedict solution, heat to boil or warm to boil, if that can happen. And the observations for X is there is no color change, so there is no non-reducing sugar absent. And for Y, you have green, yellow, and finally orange color changes, or it can change up to brown. Reject wrong sequence, but I want final color. So if you say color changes to orange, you give a mark. But if you commit yourselves to write the series, that is from blue to green to yellow to orange, the sequence must be correct. And therefore the conclusion is that non-reducing sugar present. Question 3, Roman 3. The solution recommended for a person who needs an immediate source of energy is solution X. And the reasons are as follows. Glucose provides immediate source of energy. Next, glucose is main source of energy, and the cell converts this molecule to energy within its organelles, or organs of the cell. Organelles are organs of the cell. Another reason is that glucose is easily used by the body without needing to be broken down or retrieved from another area of the body. And also, glucose is the major free sugar circulating in the blood of higher animals, and the preferred fuel of the brain and the nervous system, as well as red blood cells, or what we call erythrocytes. And finally, the reason is non-reducing sugar have to be hydrolyzed to reducing sugars to be used as a source of energy. May I now conclude this tutorial, or you can call it a lesson. Through this practical, we have seen how plant and plant cells differ in structure, how gills enable gaseous exchange in fish, and food tests help detect essential nutrients. Through this tutorial, students will gain confidence in identifying cell structures, understanding respiratory adaptations, and applying biochemical tests. These skills not only prepare learners for exams, but also foster a deeper appreciation of how biology connects microscopic details to real-world functions. Understanding these basics strengthens your grasp of biology and prepares you confidently for lab exams and real-world application. Keep exploring. Every experiment brings life to what you learn in class. So I'll finish by saying that this tutorial was made possible through the collaborative spirit of educators and learners and creators across East Africa. Special thanks to students and teachers from Kenya, Uganda, and Tanzania who continue to inspire practical excellence. Visual and conceptual support from the Smart Traveler Memos Initiative, Blending Science with Storytelling. Creative collaboration with Microsoft Copilot for adaptive visual design and poetic overlays. EACE curriculum pioneers who uphold clarity, curiosity, and culture relevance in science education. And finally, credits to every slide, specimen, and solution